Hello and welcome to the second day of our workshop on using Earth observation for water quality monitoring, and thank you very much for joining us today. My name's Paula Lightfoot. I'll be the chair for today's session. I work as an Earth observation specialist at JNCC, and for those of you not familiar with JNCC, we're a public sector body and we advise the UK government on matters of nature conservation. That's both UK-wide and internationally. For those of you who are joining us for the first time today, here's a bit of background to the workshop. We're actually running this event as part of a wider JNCC project called the Copernicus Project. And this is funded by the European Commission under the Caroline Herschel Framework Partnership Agreement on Copernicus user uptake. And that was set up a couple of years ago with the aim of increasing the use of Copernicus data, products and services. Our particular Copernicus project is aiming to increase the use of Copernicus data and services across the UK, and through doing that, enabling delivery of public environmental functions more effectively across multiple policy areas. We're achieving this through a set of work packages which we're delivering in collaboration with a large number of partners, and this is including things like capacity building, training sessions, R&D to support new practical applications, and of course delivering events such as this workshop. The aim of today's workshop is to raise awareness of how Earth observation can be used for water quality monitoring. And we've got a particular focus on how EO can be used for fulfilling environmental policy requirements in this area. Just in case anyone hasn't accessed these yet, we've got a few resources about the workshop available to download from our website, and they're also on the Eventbrite event page. So you can download the programme for the workshop, also information for attendees on how to participate, and then a selection of useful links to a wide range of sources about information on using EO for water quality monitoring. Before we go any further, I've just got a little bit of housekeeping to help the workshop run smoothly. So as yesterday, this session is being recorded, but attendees will not be visible in the recording. The videos and the slides will be publicly available on our website, and we had quite a few requests about this yesterday. There's some brilliant slides packed with information. We will be sharing them, and we'll try and get those on the website next week. We'd like to ask you to just follow these few guidelines throughout the workshop. So please keep your microphone muted and your webcam off, unless you're actually a speaker who's presenting. And we're not using the GoToMeeting chat for asking questions of the speakers about their talks. We're actually using Meeting Pulse for that. And I'll give a demonstration of that in a minute. But of course, please be polite, positive, and constructive in any questions you do ask. We would love you to tweet about the workshop. I know quite a few people were doing this yesterday, so thank you very much to them. If you do tweet, please use hashtag JNCC for water, and you can tag us at JNCC UK. And then finally, at the end of the day, we'd be very grateful if you could fill in our feedback form. It's a very short online survey, and this will really help us when we're planning future events. All feedback is given anonymously. My colleague Emily is going to put a link to the survey in the chat function in GoToMeeting, and you'll also get it in an email from Eventbrite. So this is what our agenda for today looks like. Yesterday we had some fantastic talks about a wide range of marine applications, and today we're moving on to look at freshwater applications. This will then be followed by a session on future developments, which looks at new projects, some innovative technology. And at the end, we've got a panel discussion, which I'll say a bit more about shortly. So as you can also see, there's time allocated at the end of each group of talks when the speakers will be very happy to take questions from the audience. You can ask your questions at any point during the workshop. I'm sure many of you will be familiar with this from yesterday's session, but for those joining us for the first time now, I'm just going to run through it. So we're using Meeting Pulse to manage questions. This is open already, so you can join it at this link here if you haven't already joined. And to ask a question or see what questions other people have already asked, you just click on the Q&A icon at the bottom of the page, then click the green Submit New Question banner, then type in your question and click enter. And please make it clear which speaker your question is for. And this is especially important in the future developments section. We've got two speakers who both have the first name Steve, so please make it clear which one of them it is for, so include surnames as well in that one. And a final point, if you like other people's questions and you want to make sure they're answered, please upvote them. So you can just click on the triangle at the side of the question. We will be reading out questions to speakers during the Q&A sessions. We'll try and get through them all. We didn't quite manage that yesterday because there was a lot of good questions. And we'll be starting with those with the most votes. So upvoting is important. Now, I mentioned that a key aim of this workshop is to increase the use of Copernicus data and products to support environmental policy. And at the end of the day today, we've got a session called Innovation Through to Impact, linking policy priorities to R&D. And this is going to be introduced by John Hicks from the DEFRA Earth Observation Centre of Excellence. In this section, our panel of experts are going to be discussing suggestions made by you, members of the audience, in response to these two questions you can see here. So, what are the priority water quality monitoring applications to meet UK or EU policy requirements? And this could be ones that you've heard about already, and yesterday's talks or today's talks, or it could be something we haven't covered in the workshop. So what is important to you, to your organisation? And then also, if you know this, what research and development is needed to make them operational? Where are the gaps in knowledge? Some of these have been suggested by speakers so far. So you can go into that and have a look at what's been suggested, upvote it, and please do make your own suggestions. We're very keen to get as many suggestions as possible. And we have had some people, I think there's about 8 suggestions in there so far. Some of them are general questions on this topic as well. So please feel free to just ask questions, even if you don't have a priority that you want to suggest, feel free to ask a question. You can see who the panel members are, so who you're posting your question to. Any of these suggestions that we don't manage to cover today will still form part of the workshop report and future ongoing discussions. So you can use Meeting Pulse to make these suggestions as well as to ask questions. And for those joining for the first time, here's how you can do it. So in Meeting Pulse, it's the same URL to join as for the questions. Click on the Ideas light bulb icon at the bottom of the page. Then you actually have to click on the question at the top of the page, and that will bring up all the suggestions made so far and the opportunity to submit your own. Then click on the yellow Submit Your Suggestion banner, type in your suggestion and press Enter. As with the questions, all these suggestions are made anonymously. On the other hand, if you want to mention your organisation name because that's relevant to your suggestion, then of course you're welcome to do that, but otherwise they will be by default anonymous. And a final point, as with the questions, please do upvote other suggestions that have been made if you want to make sure they are discussed by the panel. Okay, so thank you very much. If you have any questions about this, about the technological side of using this, please do put them in the chat and we'll try and help. We're now ready to start our session on looking at freshwater applications, and I'm going to hand over to our first speaker, who is Professor Andrew Tyler from the University of Stirling, and he's going to talk to us about the Globo Lakes project. Over to you, Andrew.
Great, very good afternoon. Thank you very much, Paula, and also Emily for allowing me to kick off this session and just to talk about the Globo Lakes project. Globo Lakes was a 5-year funded project by the Natural Environment Research Council. In fact, it probably lasted 7 years, and its legacy lives on, as I think if you can look at the authorship here in front of you, you'll see many of the names reappearing in this afternoon's program. So Globo Lakes, I think, was very much a very important project in the way it actually transformed our ability to exploit Earth observation capability from looking at individual lakes to the global scale. And it was only able to do that really through collaboration and bringing together the global community of Earth observation scientists, remote sensing scientists working in inland and other more optically complex environments, fundamentally sharing their data. Globo Lakes really was conceived around the concept of lakes as sentinels for environmental change. And really then ask the question, well, what is the state of lakes globally and how are they indeed changing and what are the drivers of those changes? So whilst there are fundamental ecological questions behind Globo Lakes, you can see by the proportion of the authorship here that a large portion of the work was really focused on the development of the Earth observation capability. Fortunately, and coincidentally, at the same time as Globo Lakes being funded, a number of other projects were also funded by the European Space Agency, in particular the Diversity Project, which was led by Carsten Brockman, who we heard from yesterday, as well as another EU FP7 project called GLASS, which has a similar remit, which was led by Steve Peters at Water Insight in the Netherlands. And then during the project, we also had additional resource coming in from another FP7 project called Inform. So we also supported by some excellent statisticians dealing with really large data sets from University of Glasgow, and we also had a freshwater ecologist at Centre for Ecology and Hydrology that supported the interpretation of this work as well. So just very quickly, by way of a quick introduction to give you the context, I'm sure we all appreciate that our inland waters give us fundamental ecosystem services to help us to function. But the increasing demands on those ecosystem services are coming under increasing conflict. Those conflicts result in deteriorations, for example, in water quality in many cases. Those deteriorations of water quality then also impact on the functioning of those systems, impacting on the biodiversity of organisms within these environments, but also fundamentally potentially impacting on the future security of the provision of those key fundamental services from these environments. All this is also being impacted by climate change as well. Now, if we look at the potential number or the estimated number of lakes around the world, you can see there's quite a substantial number of lakes globally. But if we work out approximately, the proportion of those lakes actually we have information on, it's only a fraction, a very small fraction of that total number of 117 million lakes globally that actually we have intelligence on. So that then really begs the question, we have a fundamental ecosystem, which we're all very dependent on, but in fact, we know very little about it. So the global lakes, then obviously we tackled this problem from an Earth observation perspective. And if we just have a quick snapshot at the sort of publications that are coming out midpoint during this project, you can see the vast majority of those publications are very much focused on individual or a limited number of water bodies. And what we found was that if you take those algorithms that are developed for these water bodies and extrapolate them to other water bodies, more often than not, they failed. Indeed, they often failed as well if you applied those algorithms sometimes to the same lake at different times of the year. And that really demonstrated that, in fact, algorithms only performed well under certain dynamic ranges of the in-water constituents of chlorophyll, total suspended matter, color dissolved organic matter, for example. So we really needed to transform an approach which was rather on a lake-by-lake basis to one which was really conceived around a pixel level and classifying that pixel according to what we're now calling an optical water type, a dynamic range over which there is certain variations or covariations of in-water constituents. So then how many optical water types are there if we're looking at lakes at a global scale? And really that was the first part of this challenge, is really to estimate that. So in winning this project, I convinced the University of Stirling to fund and build a boat purposely for this project to enable us to deploy our sensors for characterizing the above water radiometry, so looking at remote sensing reflectance above water, so we can compare that data with satellite data and compare against atmospheric correction, for example. But also characterize the in-water environment as well, the biogeochemical properties and biooptical properties of the in-water environment, so we can understand the absorption and scattering characteristics of different in-water constituents as well, and help use that to feed appropriate algorithms for that extraction of that data. So we had a boat built, which enabled us to actually shuffle that boat around the UK and put it into a whole range of different types of water bodies, inland water bodies, lakes and reservoirs in particular. But obviously getting a boat across to Europe to other lakes is difficult, so we had to borrow boats and ships, in fact, across Europe, working with partners around Europe to add to that database. And the team worked exceptionally hard to collect data, both in terms of space and time, for around 30 water bodies, lakes and reservoirs across Europe. But 30 water bodies to then help validate a project which is looking at 1,000 lakes around the world is clearly not enough in terms of understanding that dynamic range. And the real change here was actually Global Lakes really galvanized that global community of Earth observation scientists to actually willingly provide their data to support this project going forward. And in fact, we over the course of the project, managed to get about 60,000 measurements, for example, of chlorophyll for 1,500 lakes around the world. Not all the lakes are in our target population for this project. But also critically, radiometric data for 250 water bodies as well around the world as well. So that provided a really phenomenal data set, much greater than we had proposed in the proposal initially, which then set challenges actually for us in terms of storing that data, but also making it available to the community. And we finally delivered that through a database which we call LimNADES, which I really do encourage everyone to look at. And if you have data to share, we are still very keen to have data and to share it also with the wider community to help push this technology forward. Now, if you look very quickly at the data, the table on the right-hand side here, you can actually see, actually we have a data set now which covers a huge range of in-water variables, but also dynamic range within those variables. So that enables us then to test algorithms really to the breaking point, identify when they operate and perform optimally. So back to the optical water type story, we took the data that we had in remote sensing reflectance for which about 4,000 measurements across 250 lakes. And again, with our statistical colleagues at the University of Glasgow, identified basically 13 optical water types against which we could then essentially provide a framework to test those algorithms. Now, what was particularly important here is, along with these spectral measurements, we also had measures of the in-water constituents and the absorption characteristics of those in-water constituents. So we can actually identify what those water bodies were in terms of their properties, in terms of their optical properties, but also in terms of their in-water constituents as well, to provide that ultimate interpretation. So just to give you an idea of how optical water types vary, so we used the optical water type algorithms that we developed, applied it to MERIS data just to see how the optical water types vary. So here's a lake, for example, here in Kyrgyzstan, where you can see, actually in this particular case, the optical water types don't vary that much, both in terms of space and time in that particular lake. So an algorithm perhaps developed for that lake might be reasonably robust. But if you go to another one, such as the one below at Asselmeer in the Netherlands, you can see the optical water types vary dramatically throughout the year. So no one single algorithm would potentially perform well in this particular system. So really emphasizes the need to actually take an optical water type approach to algorithm application rather than the whole lake system approach. And another example here on the right-hand side, you can see how the optical water types, in this case in a Cambodian floodplain, varies with time. So you can see the color changes there reflect changes in optical water types, and in fact also this lake disappears as an ephemeral lake and then with rainfall then fills in again. So that provides a really solid foundation then to take this forward as an approach to actually then applying Earth observation at the global scale. So as part of this, then we also validated or assessed and validated atmospheric correction algorithms, which are listed here on the left-hand side, including polymer, which just became available, so enabled us to test that algorithm, which actually became one of the best performing algorithms, as has already been mentioned by Schubert, for example, yesterday. And then we also tested a whole range of in-water or biogeochemical algorithms for extracting biogeochemical variables, including chlorophyll, Phycocyanin as a pigment for Cyanobacteria, harmful algal blooms, but also total suspended matter and color dissolved organic matter. And I'm just going to focus here, given the time, just on chlorophyll, and I've just listed here the published algorithms that we then used and tested against these optical water types, and they included empirical algorithms, neural network algorithms, and analytical algorithms, for example. So that then provided a framework for us to develop this work. So we had our optical water types, and then we ingested the data that we had within the LimNADES database. But also then from the data that we had in situ, PML provided the matchup data in terms of the MERIS matchups, so that we could then test these algorithms both on in situ data, but also on actual satellite data as well. And what we did basically then was take each algorithm and identify which ones perform best, both across the whole range of data, but also ultimately identify which algorithms perform best within each optical water type. We then retuned those algorithms within each optical water type to ultimately provide that final way forward in terms of the processing that we then implemented, and that is very much that per pixel application of an algorithm according to the optical water type that we have. And then we have a scatter graph there on the right hand side, which has been published. So you can look at the paper from Neil et al. 2019. Neil is another co-author on a paper coming up this afternoon, which just demonstrates that procedure and the results in much more detail. What we found was that basically with the in situ data, 6 algorithms were very effective in covering the 13 different optical water types that we had. We did the same also with the MERIS matchup data as well. So we have here a comparison to graphs. One on the left-hand side here really uses the most relevant algorithm applied to a given lake and compared it with obviously in situ data. And on the right-hand side is the new processing technique that we've developed and now processed by PML, and that's using a blending approach, so a per pixel approach where basically we used a blending approach using a membership function and blending the results basically from the 2 closest matching optical water types for each pixel. And the result there you can see is a significant improvement in the overall response, but also the dynamic range in that response for the processing that we're doing. Also importantly, we identified algorithms which performed across different optical water types, so we could then produce optical water type families. So for example, optical water type number 1 here really represented, so optical water type family number 1 here represented essentially optical water types which were either clear or with increasing color dissolved organic matter. The next one, optical water type 2, which includes, sorry, optical family type 2, which includes optical water types 2, 8, 11, and 12, for example, are more productive waters with increasing chlorophyll and increasing color dissolved organic matter. And 3 were similar, but increasing chlorophyll and increasing total suspended matter, and 7 was really very highly productive waters. Okay, so I'm going to run out of time, so I'm going to move on very quickly. So the optical water type approach also provides a really robust framework for assessing uncertainties. So if we look at a pixel class membership function, so the probability of a pixel aligning with a particular optical water type, the higher that probability, the lower the residual error basically in that chlorophyll concentration estimate identified here on the right hand side. It's also now being used and driven by PML forward in the Lake CCI project, which I think we're going to hear a bit more about later. Also for looking at the uncertainties associated with atmospheric thickness above a pixel, and also the adjacency effects that we might also experience in the work we do. So just very quickly moving on. So this has then built a processing chain, which again was a collaboration between Brockman Consult, Plymouth Marine Lab, which has now operationalized that processing chain, and Sterling in terms of delivery of the appropriate algorithms for that. And that processing chain was then used to process 1,000 lakes globally for the MERIS time series of data for 10 years, and has subsequently now been developed also to support other European projects such as EOMORES and Monocle, again, which we'll hear more about. But also, as we heard about yesterday, it supports the Copernicus Land Service data products that Carsten spoke about as well. And then I think we'll hear more about Lake CCI and SOTO as well this afternoon. So very quickly now, just to demonstrate a comparison between in situ data and satellite data. This is one of our pet lakes, which is Lake Balaton in Hungary. And you can see here the comparison between satellite data in red and the in situ data, which is collected by our colleagues at the Balaton Limnological Institute in Tihany in Hungary. And you can see that when the biomass, the chlorophyll concentrations are reasonably low, there's a very good comparison, in fact, between what we're observing between the satellites and what's being sampled. But as the spring bloom or the summer bloom develops, you can see, in fact, the satellite data appears to underestimate what's being sampled in the water. And it's really important to have a correct interpretation of this when we see this. Because what we have to remember is that the satellite's measuring very much what it can see at the surface, particularly in these very turbid waters in Lake Balaton. But the sampling procedure uses a 2-meter tube. So a 2-meter tube goes into the water, samples the water, and then it ends up in a bucket for analysis. And what that is doing is actually sampling a subsurface maximum of chlorophyll, which we can't necessarily see from space. So it's important to have that interpretation just to understand the differences between what we can see from space and what a particular institute might be doing in terms of their sampling, so we get an appropriate comparison. So very quickly again, a quick summary in terms of what the results look like. So this is 1,000 lakes globally, and here we can demonstrate really the ecological status of these lakes as a snapshot in time. So those 1,000 lakes are the largest lakes, generally speaking, around the world, and they represent about 50% of the world's surface fresh water. So here we have a real assessment of understanding what these lakes are looking like. And we can actually proportion these lakes up in terms of those that sampled to identify that ecological status according to different parts of the world. So if we look at Europe, for example, we do seem to have a bigger proportion of lakes which are either eutrophic or hypereutrophic. We have a time series of data, and again, our statisticians have helped us look at this data, cluster it in particular, so we can look at global trends of how water quality is changing. And this is in this case, obviously, for chlorophyll. So the orange and the yellow blobs here represent actually lakes which aren't changing that much in terms of chlorophyll concentration. The blue dots basically showing water quality that's improving, chlorophyll concentrations are decreasing, and the green conversely is where basically chlorophyll concentrations are increasing. We also had a team of scientists at the University of Dundee which also looked at the catchment scale data to enable us to understand these changes that we're observing in lakes as well. And they worked really hard to collect data for what actually turned out to be 25% of the world's land mass, which is not insignificant. And they derive sensitivity factors for these lakes, i.e. the morphology of the lakes, the depth of the lakes, the geology of the catchments, those factors which aren't really changing that much in time. And then also those variables which do change in space and time, so climate variables, air temperature, rainfall data, for example, how that's changing, and included also other variables such as population change, land cover change, increasing agricultural land, for example, or urban areas, for example, and use that basically then to help identify what are the drivers of change that we have. And this is a preliminary analysis of that data that we have now, and hopefully will be published fairly soon. So what we can see here, that lakes generally increasing in chlorophyll, well, no surprises, that there is a really strong link there with nutrient concentration, but also temperature, latitude, position of that lake, percentage agriculture and urban land within the catchment. Decreasing chlorophyll, very much associated with the area of the lake and the depth of that lake, but also variations in rainfall. Equally, you can identify what each individual driver might be changing. So temperature seems to have a greater influence on deep lakes, for example, in terms of its ecological or chlorophyll concentrations, and percentage urban land seems to influence lakes in particular, which have a short residence time and water flowing through them. Okay, so I think I've probably run out of time there. So just very quickly, some key highlights. I think hopefully we've demonstrated now a really robust way of applying Earth observation data at that global scale to what we call optically complex waters. And we'll see more about this, I think, in the subsequent talks this afternoon. Also, we're now beginning to understand through this analysis, well, what are the differential sensitivity factors affecting lakes, and how can we use that potentially in management? Fundamentally, also, what we've developed is a new paradigm for monitoring water at a global scale, and indeed a regional or indeed local scale, especially in areas of the world which are data poor. So particularly in developing countries, for example, there are very real opportunities here for helping countries to basically manage their water more effectively. I should just say that also I'm now able to announce this week that there's also now a community support action that's just been funded called Waterforce under Horizon 2020, which is being led by our colleague T. Kutsa at the University of Tartu. And this project is really about working now closely with the public and the private sector to identify what their needs are and help them use that information to really develop a roadmap for the next series of Copernicus inland services. So thanks very much, everyone. I think I'll stop there.
Okay, thank you very much, Andrew, for that excellent introduction to Globo Lakes. That is obviously a very ambitious and important project. And thank you also for highlighting the links to some of the other projects we're going to be hearing about later today. That was really helpful. We're now going to move on to hear from Vangelis Spirakos, who is also from Stirling University. Vangelis is going to talk to us about using EO for monitoring cyanobacteria, and we'll then take questions for both Andrew and Vangelis at the end of this talk. So please do post any questions in Meeting Pulse.
Hello everyone, good morning, good afternoon, good evening, depending where you are. I'm sure many of us have noticed a marked discoloration like the one appearing on this first slide of water, especially when the weather has been warm. When we're visiting a lake as part of our daily lives, to walk around the lake, to cycle around the lake, to walk our dogs. Cyanobacteria are often the ones to blame for this discoloration, but also for the shutdown of local water sources and occasional death of animals. Dogs, it was mentioned as well yesterday that it has been recently in the news that elephants have been dying, and this been associated with cyanobacteria blooms in Africa. I know Peter was very eager to present this, but getting married is there on the top of good reasons for not attending a workshop. So myself, Vangelis Spirakos, I will present this on behalf of the teams at Stirling, Glasgow, CEH, and 3DO. So I will present you a way to use Earth observation for the monitoring of cyanobacteria blooms at national scale. But we know that cyanobacteria blooms occur around the world, so it will be great to hear from you. So you can pop this in the chat. Where are you calling from, and if there is any lake or lakes close to you that you know or you've seen that they have these cyanobacteria blooms. Just to finish the introduction part here, I'm a lecturer in Earth observation at the University of Stirling and program director of the Earth and Planetary Observation MSc. We now know that the main drivers of blooms are nutrients and climate, and with higher temperatures to generally favor cyanobacteria blooms. Cyanobacteria blooms occur every year throughout the UK, and if you look at the figure on this slide, this shows that increased phosphorus and temperature have generally a positive effect on the cyanobacteria abundance in Scotland, and this is based on ground data collected by the Scottish Environment Protection Agency. And we have also shown in previous research that UK lakes and reservoirs experience increases in cyanobacteria biomass due to increases in water temperature and reduced flushing. A plot here on the right is from our global change biology paper with orange cycles showing lakes where both temperature and retention were found to affect cyanobacteria biomass. And we see that most pronounced increases that are above 55 degrees latitude. But there is this lack of systematic monitoring of these lakes to be able to detect these changes in risk in order to protect our water security, but also the human health. This is an Earth observations workshop. So how can we detect these blooms using Earth observation? Let's look here at what a radiometer sees, records, and this can be on the ground or this can be on board a satellite platform, but there the detail will not be so fine. First, we see that with increasing chlorophyll A from 1 microgram per liter to 1000 microgram per liter, and going from the lighter cycles to the darker ones, that water becomes greener. and that is reflected now by the more pronounced peak in the green. We can also make use of the absorption of chlorophyll A at 665 nanometers to quantify the chlorophyll concentration. But we know that chlorophyll is common to all phytoplankton species. It will give us a really good indicator, though, of the phytoplankton biomass. Another thing we can look at is the peak in near infrared, short wavelength infrared to detect surface scams. And we can go a step further and use the absorption and fluorescence signatures at 620 and 650 nanometers respectively to quantify a phycocyanin, that is an accessory pigment in cyanobacteria. Points 1 and 2 have, and 3 as well, have greater applicability across sensors. There are many sensors out there that they record this information at this wave bands, but as we said, especially points 1 and 2, they are general for all phytoplankton species. Point 4 now is more specific to cyanobacteria. In other words, Earth observation can be generally used for detecting blooms, and specifically we can exploit phycocyanin and SCAM signatures to detect cyanobacteria. I'd like to illustrate this point by showing an example, another example coming for our pet lake here, Lake Balaton, where we used the MERIS sensor to retrieve phycocyanin. We firstly developed a retuned model for retrieving phycocyanin from satellite data. This is the top set there of the 3 figures, so the matchup of satellite data retrieving phycocyanin against the measured phycocyanin in within 1, 3, and 7 days. As expected, a better retrieval there, using same day or 3 days matchups in 7 days. Within 7 days, many things can change. When you plot this phycocyanin retrieval now against the biomass, the cyanobacteria biomass over 5 years, you can see that the MERIS product captures well the peaks in the cyanobacteria biomass. And if you apply this algorithm to a series of satellite images, and this is on your right here, you can see how cyanobacteria biomass builds up towards the end of August, especially in the western basin of Lake Balaton, something that we know because this is the site of Balaton that's closer as well to nutrient inputs. But most the lakes here in the UK, they're not like Balaton. They are not these big lakes, and bloom monitoring in the UK faces some rather specific challenges. First of all, most of our water bodies cannot be observed at 300 meter resolution, and this is what is offered now by sensors such as MERIS and OLTSI. There are higher spatial resolution sensors, but these lack a band at 620 nanometers, as we previously saw needed for detection of phycocyanin. We have less frequent revisit cycles offered by other mostly higher spatial resolution sensors, but these they have a reduced number of images in defined time periods, and also we have the cloud cover that further reduces this number of available images. But we know there is a plethora of past and current sensors with suitable or yet unproven capabilities for chlorophyll A quantification, and mostly no or proven potential for phycocyanin quantification. And this table here nicely summarizes these sensors used for chlorophyll A and phycocyanin. So what is the solution? I front-loaded this with a lot of challenges, but how can I actually tackle this? When Well, now what we can do is develop chlorophyll A products using multiple sensors and predict the likelihood of cyanodominance using a priori knowledge, such as, for example, the type of lake and the date. And I will show you how we do this. Moving into the future, we will develop methods for phycocyanin detection from future missions with high resolution orange bands, that, as for example from Planet Labs Super Dose. And this is how the current solution looks like. We use a sensor with a relatively fine spatial resolution that actually can observe these smaller lakes to calculate chlorophyll A. You can also see here the threshold defined by the World Health Organization for moderate risk. Of course, we are going to have some gaps created by clouds, but we can use other platforms like Landsat and Planet Doves to fill in some of these gaps, but also it can create data from automated in situ sensors and information collected by citizens to validate this data, but also increase the dimensionality of the service. Of course, we need to include several interconsistency tests when we have this multi-sensor approach. And then we can build a probabilistic model of cyanobacteria dominance. And we can say, for example, in spring, there is 20% probability of cyanobacteria dominance, whereas in summer, the probability goes up to 90%. I want to turn your attention now to the UK Lakes Observatory. This is a pilot service that builds on NERC-funded research that we have carried out over these last 7 or so years, and the pilot itself is funded from the NERC Climate Resilience program. This service provides chlorophyll products for 933 water bodies in the UK every week at 20 meters resolution using a dynamic selection of algorithms that Andrew mentioned earlier, this time specifically for tuned for the UK, and I think Claire as well will go into further detail after the break. We provide the data to end user via a user-friendly Ethernet-based platform built by 3DO. The link is not live currently, but we'll roll it out soon. So please note this link down. I have a video here. I hope it will play with no problems. Seems that it's doing okay. So this is a teaser here to give you an idea of the visualization end of this service. We have currently 933 lakes in the UK, and these numbers inside the cycles show the number of lakes in that area that they are included in the UK Lakes Observatory. So if you zoom in, you will see the individual lakes. Another interesting feature here are the filters that you can see on your right hand. You can use this by sliding right to select lakes that they are categorized as eutrophic, mesotrophic, oligotrophic, etc. But also there is another set of filters. I'll try to pause this. There's another set of filters to see lakes that are improving, not changing, deteriorating, or highlight the ones that they show bloom conditions. So there is a series of statistical calculations using the Earth observation data, where the current Earth observation data compared to the last 4 weeks data, and using some a threshold and percentiles. You can, of course, go and select your lake. For example, here another of our pet lakes is Loch Leven, and you can specify here the bars. So if I pause it again, yes, there it is. So the bar plot show chlorophyll A in previous years. You can, of course, you can as well change you can move them up features among different layers, as you can see here as well. But also you can go and select the data, and this will bring up the satellite image from this specific day. This goes back to 2016 and up to the last week's data. There are some other features. You can go, for example, there are URLs. They're connected to UK CEH portal, and you can go and see the monitoring data from this from UK CEH. So let's focus here on one lake, for example, Esthwaite water in the Lake District, and monitor the monthly changes in chlorophyll A using the Sentinel-2 data. We see a typical pattern with lower chlorophyll A concentrations during the winter and higher during the spring-autumn time in most of the cases. I'm aware that as well I'm over time, so I will try to go quickly through these slides. When we plot now this against the in situ mean data, in situ data provided by UK CH, we see that the satellite data, the chlorophyll A retrieved by Sentinel-2, generally follow the pattern that we see with the in situ data. You have also the dotted lines is the average, the year average, using the in situ data. This is in red against what's been calculated using the Sentinel-2 data. Comparing now to the 2020 concentration of chlorophyll A in this specific water body to previous years, we can see a decrease, a pronounced decrease in chlorophyll A during 2020. This is more apparent and statistically significant in April, and could probably show a positive effect there of COVID-19 lockdown on water quality. But this is something we further investigate at the moment. We've seen other examples from Venice Lagoon where we noticed, where our colleagues there noticed an improvement in water quality associated with COVID-19 lockdown. There were a couple of questions yesterday on Planet Labs DOV data, so I sneak in here a couple of slides in this presentation. This is again from Loch Leven. We see a nice image from Planet Labs, and you can see there some what it looks as cyanobacteria blooms in the zoomed in part image. We ran some preliminary analysis on these images on a series of Planet Labs DOV images using the normalized difference chlorophyll index that seems to have a sensitivity to biomass. We also notice issues with sensor calibration, atmospheric correction, cloud and shallow water masks is something that we are working at the moment, but there is a potential of the use of this data for detecting the biomass blooms, but maybe not in a quantitative way. And yeah, I'm reaching out towards the end. I think this is almost the last slide. Ongoing efforts now include the integration of citizen science monitoring, and especially the UK CEH's blooming algae app, but also integration with a wireless sensor network. And this is something that I think Andrew will present later. This is a monitoring buoy that we have in Lake in Loch Leven. Sorry. In conclusion, let me sum up some of the main points and next steps. The UK Lakes Observatory will launch in November, December this year for a 12-month pilot phase. The satellite lake chlorophyll data will be distributed as well via through the UKCH app and portal. We're working on further improvements to current algorithm. We've seen this something we've seen during the last year as well, that in low chlorophyll A waters, most of the algorithms they don't perform so well. So we are working towards improving the algorithms for that end of chlorophyll A, and we will keep working on the Bayesian models for predicting the probability of cyanobacteria dominance, and we are going to have this applied in several selected case studies. And I think that would be me. Thank you again for listening.
Okay, thank you very much, Vangelis. That was excellent talk. And thank you also to Andrew for the talk that came before. We have gone a little bit over time, but what I would like to suggest, I've seen quite a few good questions have come in for both Andrew and Vangelis, is that we start taking them. We'll go into the break time a little bit. If you need to leave, any individuals, obviously you can do, but please come back promptly at 2:05 for our next talk from Claire. But Emily, would you like to let us know what questions we've got? And Andrew and Vangelis, if you'd like to put your webcams on, as I think Andrew that's in the process of. If you like, then we can, yeah, perfect, thank you.
Brilliant. Thank you, Paula. Yeah, we've had some fantastic questions come in, so thank you for submitting, everyone. I'm going to start with a question for Andrew, which is, do you think the 13 optical water types are representative of all lakes globally, or might you find more than 13 types as you do more sampling?
Good question. Sorry, just yeah, I'm unmuted. But I think the first thing to say is that the 13 are essentially an artificial construct of statistics using a k-means classifier type approach. So that you need to bear in mind first of all. And the 13 were basically those that we thought were optimal. So ultimately, there could be more, and there's obviously a function of the data that we have. And hopefully we will get more data, and as people will volunteer more data, we can obviously revisit that. But I think ultimately what matters here really is the application of the algorithms and the range of optical water types over which those algorithms perform. And I think what we have here is a reasonably robust way forward insofar as, for example, with the MERIS data, we know that there are 4 optical water type families, for example, over which those algorithms seem to perform reasonably robustly. There's still more work to be done, and I think that's got to be the key point here. And we still need more data, clearly. And there are challenges, particularly at the lower end of the spectrum, in terms of very low concentrations of optical water types, low concentrations of chlorophyll, for example. And we have a PhD student between PML and Stirling tackling that question in particular. So yes, there could be more, but it's really the interpretation of those and what they mean in the application of the appropriate algorithms for extracting those important constituent data that we need.
Yeah. Okay. Fantastic.
Can I add something there as well? Do you mind?
Please do, Vangelis. Yeah.
So yeah, this is the 13 optical water types, as Andrew said. It was based on what we had at that moment, and that was quite representative. So we had a few hundred lakes there in our data set. And what we've seen as well, that when we go and try to match a new lake to a new condition to optical water types, we get a weight. We know how well this matches what we have already in our list of these 13 optical water types. So what we can do, what we are doing actually as well, is seeing lakes that they have very low score there, that the weight there is the distance is very low. So we can go and add optical water types, and we've done it for specific cases in New Zealand, for example, where we've seen lakes there that we didn't have in our optical water types. But it is the framework, that is a framework that we can add and remove if we need optical water types. And we've seen as well, like, not only lakes, we've tried it in coastal environments, in lagoons, in rivers, and we've seen that they're quite representative.
Yeah,
It's
interesting.
worth mentioning, sorry, just worth mentioning the work you did in terms of incorporating other data sets, for example, coastal water data and the optical water types you came up with there, which has been published.
Yeah, so we have another set as well for optical water types, which is more coastal waters. But in many cases, we've seen that for near shore waters, the lake optical water types, they perform better. Maybe they capture more of the variability there, but maybe because it's so close as well to land, that we get like quite an influence there from the particles from the land. But it's been like we're applying it in Venice Lagoon, and we found their optical water types that appear there as well. So.
Yeah, fantastic. And I wonder if recently I've seen quite a few Earth observation projects that have looked at the water quality of the Venice Lagoon and how it's changed over COVID-19 and the kind of clarity that we're seeing there now. So yeah, wonder how that has had an impact as well. Just linking on from that and thinking about algorithms, Andrew, what was it that made polymer better than other atmospheric correction algorithms?
Well, I'll answer this, but I'm forgetting, so others might want to come in here as well. I think the critical thing here is that polymer preserved the shape, the spectral shape, better than most of the other responses. Whilst we didn't always necessarily get a one-to-one relationship between what we got from space and with the in situ data, the overall shape was preserved much better than any other algorithm. And I think that was the key driver to really take this algorithm forward. Is there anything else you want to add there?
No, that's it. We tested it. We tested a number of different atmospheric correction models. It was a quite a wide list of the available atmospheric correction models that we had at the time, and the polymers seem to perform better than other ones for MERIS data, especially when retrieving chlorophyll A. For CDOM, for other parameters, we reconsidered that a little bit as well. We thought that other algorithms they provide, they might different atmospheric correction models, they might provide more accurate values in the blue, for example. But overall, with the data in situ radiometric data that we had, a polymer seemed to perform better. And this is now, there are a few papers out in the last couple of years in remote sensing of environment, remote sensing, that they show that a polymer is one of the candidates for atmospheric correction over these waters.
Brilliant. Okay, thank you very much. I'm just going to take our next question to yourself, Vangelis, and it is: how should members of the public report cyanobacteria blooms other than right now in the GoToMeeting chat function? For example, in England, would they report to the Environment Agency? And you have mentioned blooming algae, which is CEH's citizen science app, but what would you recommend?
I think blooming algae is used widely, and since it's out there, I think it's a good tool for the members of public to go and report report the blooms. They can upload images as well, if I remember well, and they can also note like latitude and longitude if they want to.
Okay,
Can
fantastic.
I just also just mention that Stefan Simmis is giving a talk this afternoon on monocle, which is also very much a citizen science orientated project, so we can pick up on that later.
Brilliant. Paula, how are we doing? Do you want to have a few moments break or another question?
Well, should we do one more question and then we'll take a very short break, because we will then want to start promptly at 5 past with Claire Neal's talk.
Okay. So for Vangelis again, the table that you showed, I think it was about halfway through your presentation, it summarized the suitability and cost of different sensors for chlorophyll A and phycocyanin detection specifically, and it looked really useful to our audience members, and they're wondering if that's published anywhere and if it's kept updated.
It's not published anywhere, but happy to share it with everyone here. We try to update it every so often when we have new sensors, but it's not published anywhere. But happy to have this shared. We have it in few reports in the past, but this is a little bit that might be dated now, I think, because they're 2 or 3 years old. But this is up to now, so happy to share this.
Yeah,
Thank you very much.
That should be available on the slides, would it be?
It will be available on the slides that we'll share next week. So it did look like a very useful research. Nice to have that at a glance comparison of suitability and cost implications from all the different sensors. And as we've talked about the Planet Dove sensors, it was good to have that included as well. Right, so we will take a very, very short break if people need to step away just for a few minutes. We'll be starting promptly at 2:05, hearing from Claire Neal from SEPA about monitoring eutrophication. So we'll be back very soon. Thank you.